Hello, everybody. Welcome back to our Chapter 5 discussion. We are now going to spend a few minutes on fermentation. So we just got done covering all of the stages of cellular respiration, that complete oxidation of glucose into carbon dioxide and water while producing 38 molecules of ATP in a bacteria, in a prokaryotic cell. But not all cells need that much ATP, or they can't because they can't use oxygen as the final electron acceptor. So fermentation pathways are ways to allow cells to still produce a little bit of ATP to survive, but they don't do all of the complex processes of acetyl-CoA step and Krebs and electron transport chain. So on this diagram, if you can see, we have that overview, kind of half of it. So here's glycolysis, acetyl-CoA step, Krebs, and electron transport chain. That was the pathway that pyruvic acid would take in the presence of oxygen in an organism that can do that. But again, like I said, there's diversity in metabolic pathways. So we're going to focus on this. So this is right here. Here's our pyruvic acid. We just produced that in glycolysis. We got a couple ATPs, we got a couple NADHs. That still all holds true. But instead of sending pyruvic acid down the cellular respiration pathway, these cells that do fermentation have different enzymes and they can go into different outcomes. So in bacteria, in general, they take that pyruvic acid. Here is a redox reaction. Oh, let's take a look. What do we have here? So we have pyruvic acid plus NADH goes to lactic acid, that's supposed to be an arrow, and NAD plus. This is a little bit of a trick question. Hopefully you got good recognizing that pattern of redox reactions in cellular respiration. But what do we have here? We have pyruvic acid and an NAD plus goes to lactic acid and or NADH plus lactic acid goes to an NAD plus. So wherever we have our hydrogens, this is where we have our electrons. Okay? The NADH loses its electrons. So who's going to gain those electrons? Well, it's the pyruvic acid is gaining the electrons right here. So who's losing the electrons are going to be oxidized. So that means pyruvic acid is being reduced into lactic acid. So by gaining hydrogen and gaining electrons, we are converting pyruvic acid into lactic acid. Okay. So we can actually see some of those hydrogens right here. All right. So in bacteria, when they go through fermentation pathways, they take the pyruvic acid and they convert it into lactic acid. That's what Pasteur figured out way back when he was trying to help the people who were making wine, is he split out the yeast from the bacteria. So if we go over here, yeast do this other pathway. So they have different enzymes. So first, the pyruvic acid is decarboxylated. So we lose some of the CO2. So from a pyruvic acid to acetaldehyde, right? So we're losing that CO2 when we go here. So acetaldehyde to another redox reaction. Let's see if you guys are picking up on this fermentation redox pathways. So we have acetaldehyde plus NADH goes to ethanol and NAD plus. All right, so where are the electrons hanging out? Remember, they're with our hydrogens. So if the NADH has the hydrogens to begin with, and then it doesn't on the product side, right, because it went to NAD plus, it had to lose those electrons. So the NADH is oxidized, which means the acetaldehyde had to be reduced because those electrons went to the acetaldehyde to create the ethanol. And it's kind of rearranged a little bit, but here are some of those hydrogens here in the middle of that ethanol molecule. And what is ethanol? Alcohol. It's the alcohol that we consume in alcoholic beverages. And if you know home brewers, or if you are a home brewer, or if you know anything about winemaking, or if you enjoy wine or beer or any of that kind of stuff, this is the organism that allows that to happen. The sugars in the grapes or the hops or whatever the thing is that you are fermenting, glycolysis produces pyruvic acid. And in the presence of yeast, the pyruvic acid gets converted into ethanol. That's the alcohol part of your alcoholic beverages. For beer, the CO2 is kept, and that's the bubbles, that's the carbonation of the beer. In wine, CO2 gets let off, and so you don't get fizzy wine, even though there's champagne, which is kind of the same thing. But yeast produces alcohol, bacteria produce acid. And it's not always lactic acid. In this case, it's lactic acid, but there's lots of different kinds of acids that bacteria can produce. But that's the general kind of two product pathway that fermentation can take. So take a look at some products. What can various microorganisms do with these fermentation pathways, and maybe some things that we are familiar with. So here is our quick, if glycolysis was only one arrow simple, but it's showing you that NAD plus gets reduced into NADH in the process of glycolysis, producing the pyruvic acid. So some organisms of the Propionibacterium genus probably ferments the sugars or whatever it's fermenting with into carbon dioxide and propionic acid, yay, which makes Swiss cheese. Cool. Bacillus, lactobacillus, and streptococcus bacteria produce sugars to make lactic acid, which can produce fermentation products of cheese like cheddar cheese, yogurt, and soy sauce. Yay. Saccharomyces cerevisiae, that's one of the organisms you have in your kit, ferments into alcohol and carbon dioxide. So those are your alcoholic beverages. Some Clostridium species, not necessarily tetanus and botulism, but there are some non-fatal Clostridium species out there. They can ferment into acetone and isopropanol, like nail polish remover and rubbing alcohol. There are some other genus, Escherichia and Acetobacters produce acetic acid, which is vinegar. So vinegar is a product of fermentation. And again, if we're tracking those NADHs, the electrons that gained here, the electrons are actually going to all of those products of fermentation, and we make more NADH. Well, is that important? Well, sure it is, because take a look. That NADH has to come back around here so the cell can continue to do glycolysis, because this is the only part of fermentation that we make ATP. So the NAD plus gets reduced into NADH to drive glycolysis to make your ATPs. Then fermentation turns around, swaps those electrons, and produces NAD plus to allow for glycolysis to keep going. It's all very interesting. Cool little connection there. And not only do this is just bacteria's waste products. So all of these fermentation products here that we've listed here that bacteria do, that's just our waste products. We as humans have stumbled across these and said, oh, yum, yogurt and cheese and wine and beer and vinegar to some degree, and then industrial uses like nail polish remover, acetones, and rubbing alcohols, just the byproduct of bacterial metabolic pathways. All right, the last slide that I have before we jump into photosynthesis is just comparing aerobic respiration, anaerobic respiration, and fermentation. I didn't talk too much about anaerobic respiration during the electron transport chain, but it would just be something other than oxygen as a final electron acceptor. So I've listed a few things here. It does all the same things. So glycolysis, acetyl-CoA step, Krebs, electron transport chain, but instead of oxygen accepting those electrons at the very end, something else does. And it allows them to do all their metabolic pathways in the absence of oxygen. So you can kind of take a look and see what's different between the ATP production, your final electron acceptor, and whether oxygen is required or not. Okay, so that wraps up our catabolic pathways, because fermentation is still considered a catabolic pathway. And then we'll wrap up with our discussion on photosynthesis, which is an anabolic pathway. All right, I'll see you next time.